अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 7 एक्सरसाइज शॉर्ट क्वेश्चन आंसर्स सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, हाउ डू डाइनोफ्लैजिलेट्स डिफर फ्रॉम अदर एल्गी इन टर्म ऑफ देयर लोकोमोशन मेथड्स। कह रहा है कि जो डाइनोफ्लैजिलेट्स हैं वो बाकी एल्गी से कैसे डिफरेंट होते हैं इन टर्म्स ऑफ देयर लोकोमोशन। Dinoflagellates differ from other algae in their unique locomotion method, which includes, first of all, flagellar movement. Dinoflagellates have two flagella, a transverse and longitudinal one, which they use to propel themselves through the water. The transverse flagellum beats in a wave-like motion, while the longitudinal flagellum beats in a helical motion, creating a spinning motion. Next is corkscrew-like motion. The combination of the two flagella's movements creates a distinctive corkscrew-like motion, allowing dinoflagellates to move through water with high maneuverability. Next is rapid movement. Dinoflagellates are capable of rapid movement, with some species able to move up to 25 body lengths per second. Next is directional control. Dinoflagellates have directional control over their movement, allowing them to swim towards or away from light, nutrients, or predators. In contrast, other algae typically move using passive drifting. Many algae simply drift with a current, lacking flagella or other locomotive structures. Gliding. Some algae, like diatoms, use a gliding motion to move along surfaces. Amoeboid movement. Some algae, like chlorophytes, use amoeboid movement, extending pseudopodia to move. Dinoflagellates' unique flagellar movement allows them to actively hunt prey, evade predators, and migrate vertically through water column, making them well suited to their environment. Next question is, list the key components found in cell structure of protozoa. The key components found in cell structure of protozoa include, first of all, cell membrane, a thin semi-permeable membrane that surrounds the cell. Next is cytoskeleton, composed of microtubule, microfilament, and intermediate filament, providing shape, support, and movement. Next is cilia or flagella, hair-like structures used for locomotion, feeding, and sensing the environment. Next is pellicle, a layer of flexible protein-based plates that provide additional support and protection. Pseudopodia, temporary extensions of the cell membrane used for movement, feeding, and engulfing prey. Food vacuoles, membrane-bound sacs that contain ingested food particles. Contractile vacuoles, specialized vacuoles that regulate water balance and osmotic pressure. Nucleus, the control center of cell containing genetic material. Mitochondria, organelles responsible for energy production through cellular respiration. Endoplasmic reticulum, a network of membranous tubules and cisternae involved in protein synthesis and transport. Golgi apparatus, a complex of flattened sacs and tubules that process and package proteins and lipids. Lysosomes, membrane-bound sacs containing digestive enzymes responsible for cellular digestion and recycling. These components work together to enable protozoa to perform various cellular functions such as movement, feeding, and reproduction. Next question is, what distinguishes foraminifera and actinopods from other marine protozoa, and where are their protective shells found? Foraminifera and actinopods are distinguished from other marine protozoa by their, first of all, complex shells. Foraminifera have calcium carbonate shells with chambers, while actinopods have siliceous shells with a network of fibers. Large size. Foraminifera and actinopods are generally larger than other protozoa. Complex internal structure. Both groups have a complex internal organization including multiple nuclei and specialized organelles. Protective shells are found in foraminifera, calcium carbonate shells with chambers called tests which provide protection and support. Tests are usually secreted by the cell membrane and can be composed of calcite or aragonite. Next are actinopods. Siliceous shells होते हैं called frustules which are composed of silica, silicon dioxide. Frustules are usually secreted by cell membrane and have a network of fibers. These shells serve as a protective barrier against predator, provide support, and help maintain the cell's shape. They are also important in fossil record as they can be preserved and provide valuable information about ancient marine environment. Next question is, where in the life cycle of plasmodial slime molds are sporangia typically produced and what is their role? In the life cycle of plasmodial slime mold, sporangia are typically produced during the fruiting stage, which occurs after the plasmodium has reached a certain level of maturity and has ceased to grow. Sporangia are structures that produce spores which are similar to the seeds of plants. They are usually produced on the surface of plasmodium, often in a specific pattern or arrangement. The role of sporangia in plasmodial slime mold is to produce spores. Sporangia produce spores through a process called sporogenesis. Disperse spores. Sporangia release spores into environment, allowing them to disperse and potentially colonize new areas. Ensure survival. Sporangia and the spores they produce ensure the survival of species as they can withstand unfavorable conditions and disperse to new locations. Complete life cycle. The production of sporangia and spores complete the life cycle of plasmodial slime molds, allowing them to transition from diploid plasmodial stage to haploid spore stage. In summary, sporangia play a crucial role in the life cycle of plasmodial slime mold, enabling them to produce and disperse spores, ensuring their survival and allowing them to complete their life cycle. Next question is, what sets brown algae apart from other multicellular algae and where are kelps commonly found? Brown algae are distinct from other multicellular algae due to, first of all, their unique cell wall composition. Brown algae contain alginate, fucoidine, and cellulose, providing strength and flexibility. Fucoxanthin pigmentation. Brown algae have pigment fucoxanthin responsible for their characteristic brown color, complex multicellularity. Brown algae exhibit complex body structures including holdfast, stipes, and blades. Large size and complex life cycle. Brown algae can grow large and have intricate life cycle. Kelps, a type of brown algae are commonly found in temperate and subarctic regions. Kelps thrive in cooler water 5 to 20 degrees Celsius, coastal areas with rocky substrates. Kelps typically inhabit shallow coastal waters with rocky surfaces. Intertidal and subtidal zones. Kelps can be found in both intertidal and subtidal zones. Specific locations where kelps are commonly found include Pacific coast of North America, California, Oregon, Washington. Atlantic coast of Europe, UK, France and Norway. Japan, Southern Australia, South Africa. Kelps form large underwater forests, providing habitat for diverse marine life. Next question is, how do euglenoids differ from other freshwater organisms in terms of their nutritional strategy? Euglenoids differ from other freshwater organisms in their nutritional strategies in several ways. First of all, mixotrophy. Euglenoids are mixotrophic, meaning they can photosynthesize like plants but also ingest and digest prey like animals. Phagotrophy. Euglenoids can engulf and digest bacteria, algae, and small organisms, supplementing their photosynthetic nutrition. Osmotrophy. Some euglenoids can absorb dissolved nutrients from surrounding water like osmotrophs. Next are flexible nutrition. Euglenoids can switch between different nutritional modes depending on environmental conditions such as light availability or prey abundance. Lack of cell wall. Euglenoids lack rigid cell wall, allowing them to change shape and engulf prey. Presence of chloroplast. Euglenoids have chloroplasts which they acquired through secondary endosymbiosis, allowing them to photosynthesize. These unique nutritional strategies enable euglenoids to thrive in various freshwater environments, from low-nutrient lakes to organic-rich sediments. Next question is, what characteristics the life cycle of oomicotes and where are swimming zoospores typically generated? Oomicetes, also known as water molds, have a unique life cycle characterized by biphasic life cycle alternating between a filamentous diploid 2N stage and a motile haploid stage. Spore formation producing sporangia which contain motile zoospores. Zoospore dispersal. Swimming zoospores disperse and infect new host. Saprotrophic or parasitic nutrition obtaining nutrients by decomposing organic matter or parasitizing living organisms. Swimming zoospores are typically generated in sporangia, specialized structure producing zoospore usually form on the tip of hyphae. Aerial sporangia. Some oomicetes produce aerial sporangia which release zoospores into the air. Aquatic environment. Zoospores are often generated in aquatic environment such as ponds, lakes, or rivers. Infected hosts. Some oomicetes produce zoospore within infected hosts such as plants or animals. Zoospores are usually generated in response to environmental cues like moisture, temperature, or nutrient availability, allowing oomicetes to disperse and infect new hosts. Next question is, how do pianoflagellates stand out from other flagellate organisms and what is their structural resemblance to? Pianoflagellates stand out from other flagellate organisms in several ways. First of all, collar of microvilli. Pianoflagellates have a distinctive collar of microvilli around the base of their flagellum, which is used for capturing prey. Flagellum structure. Their flagellum is surrounded by a ring of microvilli, creating a conus or collar which gives them their name. Filter feeding. Pianoflagellates use their collar to capture small particles and bacteria from the water, making them filter feeders. Next is simple single-celled body. Pianoflagellates have a simple single-celled body structure lacking complex organelle. Ancient lineage. Pianoflagellates are thought to be one of the oldest eukaryotic lineages with fossil evidence dating back over 1 billion years. Structurally, pianoflagellates resemble sponges, porifera. The collar of microvilli is similar to the choanocytes collar cells found in sponges, which are also filter feeders. Animal cells. The basic body structure and flagellum of choanoflagellates are similar to those found in animal cells, suggesting a close evolutionary relationship. Early eukaryotic cell. Choanoflagellates are thought to resemble the earliest eukaryotic cells, providing insight into the evolution of eukaryotic life. Next question is, where in the life cycle of red algae are holdfasts found and how do they anchor these multicellular organisms? In the life cycle of red algae, holdfasts are found in the mature thallus stage. Holdfasts are present in the mature multicellular thallus, vegetative stage of red algae. Attached to substrate. Hold fast anchor the thallus to a substrate such as rocks, reefs or other surfaces. Hold fast anchor red algae by adhesion. Hold fast secrete adhesive substances that attach to the substrate. Rhizide-like structure. Holdfasts have a rhizide-like structure with branching, finger-like projections that grip the substrate. Cellular attachment. Cells in the holdfast region attached to the substrate provide additional anchorage. Mucilaginous secretions. Some red algae produce mucilaginous secretions that help anchor the holdfast to the substrate. The holdfast serve as a anchor, securing the thallus in place, preventing detachment by currents or waves. Support, providing structural support for thallus, allowing it to grow upright or spread out. Nutrient uptake. Some holdfasts may also play a role in nutrient uptake, absorbing nutrients from surrounding water or substrate. Next question is, list 4 different types of locomotion methods used by protists and give an example of a protist that employ each method. First of all is flagellar movement. Protists use flagella, whip-like structures to propel themselves through water. Example, trypanosomes. For example, Trypanosoma brucei use a single flagellum to move through the bloodstream of their host. Ciliary movement. Protists use cilia, short hair-like structures, to create currents and propel themselves. Example, Paramecia, for example, Paramecium caudatum, use cilia to move through water and feed on bacteria. Next is immoboid movement. Protists use pseudopodia, false feet, to slowly move and change shape. For example, amoebas Amoeba proteus use pseudopodia to move through water and engulf prey. Next is gliding. Protists use a gliding motion to move over surfaces, often using mucilaginous secretions. Examples include diatoms like Navicuola pelliculosa use gliding to move over rock and other surfaces, leaving behind a trail of mucilage. These locomotion methods allow protists to move and interact with their environment in unique way, often adapted to their specific ecological niches. Next question is, list 3 examples of parasitic protozoa that infect humans. Examples of parasitic protozoa that infect humans include, first of all, Plasmodium falciparum causes malaria, a serious and sometimes life-threatening disease transmitted through the bite of an infected mosquito. Giardia lamblia causes giardiasis, an intestinal infection that leads to diarrhea, abdominal cramp, and weight loss, typically spread through contaminated water or food. Trypanosoma brucei causes African trypanosomiasis, sleeping sickness, a serious disease transmitted through the bite of an infected tsetse fly, leading to fever, headache, and eventually neurological problems if left untreated. These parasitic protozoa can cause significant harm to humans, emphasizing the importance of prevention, diagnosis, and treatment. Next question is, what are primary modes of obtaining nutrients in fungi? The primary modes of obtaining nutrients in fungi are, first of all, saprotrophy, decomposing organic matter and absorbing nutrients from dead organisms; parasitism, obtaining nutrients by infecting and exploiting living organisms such as plants, animals, or other fungi; mycorrhizal association, forming symbiotic relationship with plant roots, exchanging nutrients and water for carbohydrates produced by photosynthesis; lichens, forming symbiotic relationship with algae or cyanobacteria, providing shelter and nutrients in exchange for photosynthetic products. Next is predation, capturing and digesting small organisms such as insects, worms, or other microorganisms. Fungi have evolved diverse strategies to obtain nutrients, allowing them to thrive in various environments and ecosystems. Next question is, where can fungi be found in various ecosystems and what are their diverse habitats? Fungi can be found in various ecosystems and have diverse habitats including: first of all, soil, decomposing organic matter, forming mycorrhizal relationship with plant and influencing soil structure. Decaying organic matter, logs, stumps, leaf litter, and other decaying plant material. Plant roots, forming mycorrhizal relationship, exchanging nutrients and water. Tree trunks and branches, growing as brackets, conchs, or cankers. Freshwater and marine environment. Aquatic fungi such as chytrids and oomycetes infecting algae, plants and animals. Animal bodies. Parasitic fungi like Candida infecting humans and other animals. Insect bodies. Fungi like Ophicordioids infecting and controlling insect behavior. Lichens. Fungi in symbiosis with algae or cyanobacteria on rocks, trees and soil. Compost and dung. Fungi breaking down organic matter in these nutrient-rich environment. Extreme environment. Fungi thriving in Arctic, desert and high radiation environment. Fungi have adapted to diverse habitat, playing vital role in ecosystem from decomposition and nutrient cycling to symbiotic relationship and parasitism. Next question is, why are fungi considered heterotrophic? Fungi are considered heterotrophic for several reasons. First of all, lack of chlorophyll. Fungi do not possess chlorophyll, the green pigment necessary for photosynthesis, inability to produce own food. Fungi cannot produce their own food through photosynthesis or chemosynthesis, dependence on external nutrients. Fungi obtain nutrients by decomposing organic matter, forming symbiotic relationships or parasitizing other organisms. Next is absorptive nutrition. Fungi absorb nutrients from their surroundings such as soil, decaying organic matter or living host. No self-sustaining metabolism. Fungi rely on external sources of energy and organic compounds to sustain their metabolic processes. Heterotrophy in fungi allows them to thrive in diverse environments, exploiting various nutrient sources and playing crucial role in ecosystem from decomposition to symbiotic relationship. Next question is, list two differences between fungal cell wall and plant cell wall. Main structural component. Fungal cell wall contains chitin, a polysaccharide polymer. Plant cell wall is made up of cellulose, a polysaccharide polymer. Next is additional components. Fungal cell wall often contain their components like beta-glucan, mannans, and glycoproteins. Plant cell wall contain additional components like hemicellulose, pectin, and lignin in some plant cell types. These differences reflect distinct evolutionary path and functional requirement for fungal and plant cell wall, influencing their strength, flexibility, and interactions with the environment. Next question is, name four major groups of fungi based on their methods of reproduction. The four major groups of fungi based on their method of reproduction are: first one is Ascomycota, also called sac fungi, produces spores in a sac-like structure called ascus. Examples: mushroom, molds, and yeast. Next is Basidiomycota, also called club fungi, produce spores on a club-like structure called basidium. Examples include mushroom, rust, and smut. Next is zygomycota, also called conjugating fungi, produce spores through conjugation, fusion of two nuclei. Examples: bread mold, black bread mold. Next is deuteromycota, also called imperfect fungi, reproduce asexually without a true cycle. Examples: mold, yeast, some species. Next question is differentiate between endomycorrhiza and ectomycorrhiza based on their interaction with plant root. Endomycorrhizae and ectomycorrhizae differ in their interaction with plant root as follows. First of all, we see endomycorrhizae. Internal colonization, fungal hyphae penetrate the plant root cell. Intracellular structure, fungi form specialized structures like arbuscles, coils, or vesicles within root cell. Direct nutrient exchange, nutrients are exchanged between the fungus and plant cells through these intracellular structures, typically associated with herbaceous plants, grasses, and some shrubs. Next is ectomycorrhizae. External colonization, fungal hyphae surround the plant root but do not penetrate it. Intercellular structures, fungi form a network of hyphae around root cell, creating a mantle or sheet. Indirect nutrient exchange, nutrients are exchanged between fungus and plant through the mantle or sheet, typically associated with trees, especially conifers and some woody shrubs. These differences reflect distinct symbiotic relationship between fungi and plant roots, influencing nutrient uptake, plant growth, and ecosystem functioning. Next question is, what makes imperfect fungi unique and why are they sometimes referred to as imperfect? Imperfect fungi, Deuteromycota, are unique in that they lack a sexual cycle. They reproduce only asexually without a true sexual cycle. No observed sexual structures. They do not produce ascospores like Ascomycota or basidiospores like Basidiomycota, asexual spores only. They produce conidia which are asexual spores, variable morphology. They exhibit diverse shapes, sizes, and colors. Next is ecological adaptability. They thrive in various environments from soil to human tissue. They are sometimes referred to as imperfect because incomplete life cycle. Their life cycle is not fully understood, lacking a clear sexual stage, unobserved sexual reproduction. Scientists have not directly observed their sexual reproduction. Historical classification. The term imperfect was used to distinguish them from fungi with well-defined sexual cycles. Next question is, what is zygospore and how is it formed? A zygospore is a type of spore that results from the fusion of two gametes in fungi, particularly in Zygomycota phylum. It's a crucial structure in the sexual production of these fungi. Formation of a zygospore. First of all, gametangia formation. Two opposite gametangia develop on adjacent hyphae. Gamete release. Each gametangium release a gamete fusion. The two gametes fuse forming a zygote development. The zygote develops into a zygospore, a thick-walled resting spore. Zygospore maturation. The zygospore matures, becoming resistant to environmental stresses, meiosis and germination. When conditions are favorable, the zygospore undergoes meiosis, producing haploid spores that germinate into new individuals. Zygospores are unique in that they are formed by fusion of two gametes, have a thick resistant wall, can survive harsh conditions, play a key role in the sexual production of Zygomycota fungi. Zygospores are an adaptation to ensure the survival and genetic diversity of these fungi, allowing them to thrive in various environments. Next question is, what is histoplasmosis? Histoplasmosis is a fungal infection caused by the dimorphic fungus Histoplasma capsulatum. It's a common opportunistic infection, especially in people with weakened immune system. Transmission. Spores. Inhaled spores from contaminated soil, bird droppings, or bad guano. Contaminated environment. Exposure to contaminated environments such as caves, attics, or soil. Symptoms. Mild flu-like symptoms, fever, cough, fatigue, and chest pain. Severe में pneumonia, respiratory failure, and dissemination to other organs, for example, brain, liver, and spleen. Forms of histoplasmosis. First one is acute, recent infection, often mild and self-limiting. Chronic, long-term infection, often in people with compromised immune system. Disseminated, infection spreads to other organs, often in immunocompromised individuals. Diagnosis: chest X-ray abnormalities in lung tissue. Blood test, detecting antibodies or antigen. Tissue biopsy, microscopic examination of tissue samples. Treatment: antifungal medication, azoles, amphotericin B, or itraconazole. Sportive care, managing symptoms and preventing complications. Prevention: avoid exposure, minimize contact with contaminated environment, wear protective gear, mask, glove, and eye protection when handling potentially contaminated material. Improve ventilation, reduce pore concentration in air. Histoplasmosis is a serious infection, especially for people with weakened immune system. Early diagnosis and treatment are crucial to prevent complications and ensure effective management. Next question is, if a new deadly fungicide is made by scientists that destroy all existing fungi, what would be the impact? If a new deadly fungicide were to destroy all existing fungi, the impact would be catastrophic and far-reaching, affecting various aspects of ecosystem, human health, and environment. Some potential consequences are: first one is decomposition and nutrient cycling disruption. Fungi play a crucial role in decomposing organic matter and recycling nutrients. Without them, ecosystem would accumulate dead plant material, leading to nutrient deficiency and altered soil chemistry. Next is loss of symbiotic relationship. Mycorrhizal fungi form essential partnership with plant roots, facilitating nutrient uptake. Without these fungi, plant growth and productivity would decline, impacting agriculture and forestry. Next is soil erosion and degradation. Fungi help hold soil particles together and maintain soil structure. Without them, soil erosion and degradation would increase, leading to reduced fertility and increased sedimentation in waterways. Next is impact on food chain and biodiversity. Fungi are a food source for various animals, from insects to large mammals. The loss of fungi would disrupt food chain, potentially leading to population decline or even extinction. Next is human health consequences. Fungi are a source of antibiotics, for example penicillin and other medicine. The loss of fungal diversity could limit the discovery of new drugs, making us more vulnerable to diseases. Economic impacts. The destruction of fungi would affect industries like agriculture, forestry, and pharmaceuticals, leading to significant economic losses. Unintended consequences. The introduction of a deadly fungicide could have unforeseen effects on ecosystem, potentially leading to emergence of new diseases or the disruption of delicate ecological balance. It is essential to consider the potential risks and consequences of such a fungicide and explore alternative solutions that target specific fungal pathogens while preserving beneficial fungi. Next question is, write two salient features of coincitic hyphae, conidia, escocarp, sporangia, and basidium. First of all, coincitic hyphae. Multinucleate coincidic hyphae contain multiple nuclei within a single cell, allowing for coordinated growth and function. No septa. Unlike other hyphae, coincidic hyphae lack septa, cross walls, enabling free movement of nuclei and organelles. Next is conidia. Asexual spores. Conidia are asexual spores produced by fungi for reproduction and dispersal. Variable morphology. Conidia exhibit diverse shapes, size, and colors depending on the fungal species. Next is ascocarp. Fruiting body. An ascocarp is a fruiting body containing acai spore-bearing structures in Ascomycota fungi. Protective structure. Ascocarps shield the acai and developing spores from environmental stressors. Next is sporangia, spore-bearing organ. Sporangia are specialized structures producing spores in fungi typically found in Zygomycota. Aquatic dispersal. Sporangia often release spores into water, facilitating aquatic dispersal. Next is basidium. Club-shaped structure. Basidia are club-shaped structures bearing basidiospore in Basidiomycota fungi. Basidia play a crucial role in sexual production, facilitating the fusion of nuclei and meiosis. Next question is, do the term dikaryotic and diploid have the same meaning? No. The terms dikaryotic and diploid do not have same meaning. First of all, we see dikaryotic refers to a cell or organism containing two separate nuclei, often of different genetic makeup, typically used to describe the heterokaryotic stage in fungal life cycles, for example, Basidiomycota. The two nuclei coexist but do not fuse, maintaining their individuality. Next is diploid. Refers to a cell or organism containing two complete sets of chromosomes. One set of chromosomes is inherited from each parent. Diploid organisms have a uniform genetic makeup, unlike dikaryotic organisms. Key differences. Number of nuclei. Dikaryotic, two separate nuclei versus diploid, single nucleus with two chromosome set. Genetic makeup. Dikaryotic, heterogeneous versus diploid, homogeneous. In summary, while both terms relate to number of genetic components, they describe distinct concepts in genetics and mycology. Next question is, how do fungi store carbohydrates? Fungi store carbohydrates in various forms depending on the species and environmental condition. Here are some common ways fungi store carbohydrate. First one is glycogen. Similar to animals, some fungi store glycogen, a branched polysaccharide in their cell. Trehalose. Many fungi accumulate trehalose, a disaccharide, as a storage carbohydrate. Mannitol. Some fungi, like mushrooms, store mannitol, a sugar alcohol, in their cell. Chitin. Fungal cell wall contain chitin, a polysaccharide that provides structure support and can also serve as a carbohydrate reserve. Beta-glucans. Some fungi, like yeast, store beta-glucans, a type of polysaccharide, in their cell. Lipids. Some fungi, like oil-producing fungi, store lipid as a carbon and energy source. Fungi have evolved these different storage strategies to adapt to various environments and survive periods of nutrient scarcity. These stored carbohydrates can be mobilized when needed to support growth, development, and reproduction. Next question is, name the four major groups of fungi based on their methods of reproduction. The four major groups of fungi based on their methods of reproduction are: first one is Ascomycota, also called sac fungi, produce spores in a sac-like structure called ascus. Examples: mushroom, mold, and yeast. Basidiomycota, also called club fungi, produce spores on a club-like structure called basidium. Examples include mushroom, rust, and smut. Next is Zygomycota, also called conjugating fungi, produce spores through conjugation, fusion of two nuclei. Examples include bread mold, black bread mold. Next is Deuteromycota, also called Imperfect fungi reproduce asexually without a true sexual cycle. Examples include mold, yeast, some species. So students, हमारे ये short question complete हो चुके हैं. Hopefully आपको समझ आ गए होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.